All the information that an organism needs is found in its DNA or its genotype. To change that information into something that can be seen, the phenotype, the organism's DNA must be converted into proteins. Proteins are the basis of life, whether they make up structures completely, like enzymes, or just large portions of it, such as membranes. Anytime you look at a living organism, at some point you move back to a protein. This chapter is about how proteins are made. This is Genes and How They Work, Chapter 15. The flow of information from DNA to protein was first described by Francis Crick. He said that information flows from DNA to RNA to the final protein. He called this the central dogma of molecular biology. Transcription is the process of converting DNA to RNA, and translation is the process of converting RNA to proteins. Together, they are called protein synthesis. All organisms but retroviruses follow this DNA to RNA to protein pattern. Retroviruses use an enzyme called reverse transcriptase to convert their RNA into DNA, and we'll talk about this later on in the chapter. Protein synthesis begins with the creation of RNA from instructions given by DNA. In this case, we will use only one strand of DNA to make the RNA. Thymine in DNA will be replaced by uracil in RNA. And what we're going to create is messenger RNA. This will be the instructions that will be used to actually create the proteins in translation. In translation, we're actually going to build the protein, also called a polypeptide. This will take place at the ribosomes, and it will require several different kinds of RNA. I'll cover each of those types of RNA as we go through translation. Before I talk about the different types of RNA, I want to look at the structure. The structure of RNA is very similar to the structure of DNA. It has a single backbone of nucleotides, unlike DNA that has the double backbone, so it's a single helix. The nucleotide is made up of a sugar and a phosphate that forms the backbone and the nitrogen base that's in the middle. The difference is that it's a ribose sugar, not a deoxyribose sugar. And the bases are slightly different. We have guanine bonding to cytosine and adenine bonding to uracil. There is no thymine in a strand of RNA. There are several types of RNA. First, we have messenger RNA. Messenger RNA is a single strand of RNA that's going to provide the information needed to create the protein. It's going to convert DNA's information into RNA, 3 nucleotides, or an amino acid at a time. These are each called codons. Codons that code for information are called exons. Codons that are used as spaces but have no information of their own are called introns. Transfer RNA is going to deliver the amino acid to its proper place along the messenger RNA strand. Each amino acid is made up of 3 nucleotides that are complementary to the codons found in messenger RNA. Because of this, they are called anticodons. Ribosomal RNA is combined with proteins from the cytoplasm to create a ribosome. This ribosome is going to coordinate the activities of both messenger and transfer RNA during translations. Ribosomes have 3 binding sites, places where the amino acids are going to be attached. One that will be used by messenger RNA, one for the last amino acid attached to the growing protein, and one for the amino acid that is going to be placed. I'll talk about this in detail when we get to translation. The last one is called small nuclear RNA. After the messenger RNA is created, these molecules will remove the introns and splice the remaining exons together. I will talk about each one of these types of RNA again as we go through the idea of transcription and translation. Transcription is like DNA in two ways. First, the nucleotides are added toward 5'. And second, it has the same process: initiation, elongation, and termination. Unlike DNA replication, we're not going to copy the entire strand. We're only going to use a small piece of it to create the protein we want. We're also going to use RNA polymerase rather than DNA polymerase to glue those nucleotides in. And finally, we're going to create a single strand of RNA, specifically messenger RNA. Transcription initiation begins with a promoter. A promoter is a base sequence in the DNA that signals the start of a gene. At this point, the DNA will begin to unzip, creating a place for RNA polymerase to bind to. If RNA polymerase isn't there, transcription can't start. The promoter then is going to serve two purposes. First, it's going to open the DNA so the work can begin. And second, it's kind of a sign that says, start here. RNA polymerase looks for that promoter and it begins its building at the sequence just immediately following the promoter. Once the promoter has been recognized, elongation can begin. RNA polymerase will bring the nucleotides in, building in the 5' direction. The site where we're actually adding the DNA nucleotides is called the transcription bubble. This is the site where you're actively working. Inside the transcription bubble is where the RNA polymerase and the nucleotides that are being added will be found. After the transcription bubble passes, the transcribed DNA will rewind. If you look at the picture, the blue is the DNA, the brown is the RNA. So we're building within that big pale blue circle the transcription bubble. The DNA opens up, the RNA begins to be built. As it builds, it leaves the transcription bubble, and then the bubble moves down the line. As the bubble moves down the line and the DNA comes out of the bubble, it will curl up and go back to its original shape, just as you see on the far left side. The last step is termination. This occurs when the RNA polymerase reaches a special sequence of nucleotides that signal stop. In eukaryotes, the DNA sequence that transcribes stop is 6 A's in a row. When it reaches those 6 A's, RNA polymerase releases the DNA, the promoter is released, and the DNA rewinds. At this point, we have created pre-messenger RNA. Pre-messenger RNA is composed of exons and introns. Exons are codons that code for information. Introns are codons that code for spaces. Those spaces must be removed. This happens before the pre-messenger RNA leaves the nucleus. This messenger RNA splicing occurs in eukaryotes only. Small nuclear RNA is responsible for removing the introns and gluing the exons together. When this is completed, messenger RNA is ready to leave the nucleolus and move on to translation. Translation converts the information from messenger RNA into the amino acids that the DNA requires. These amino acids will be glued together to create the final protein. This is going to take place at the ribosomes and will use the same steps of initiation, elongation, and termination. All messenger RNA strands are going to begin with the same codon, AUG, which signals start. Initiation is going to begin when a small piece of ribosomal RNA attaches to the end of the messenger RNA just in front of the AUG. Once this piece of ribosomal RNA, called the small ribosomal subunit, is in place, translation can begin. And in our picture, it's going to be the little tiny blue bubble underneath the big giant blue bubble. A transfer RNA molecule carrying the amino acid methionine, also known as the anticodon UAC, is going to attach to the messenger RNA at the P site. So if you look at the top picture, you're going to see the tRNA, that's the yellow circle, with a little circle that says MET. That's the P site. A large piece of ribosomal RNA, called the large ribosomal subunit, will attach to the messenger RNA on top of the small ribosomal subunit. That's going to be the big blue circle. Together, the big blue circle and the little blue circle, or the large ribosomal subunit and the small ribosomal subunit, create a ribosome. Once the ribosome has been made, elongation will begin when the next transfer RNA comes in. It's going to have the next amino acid on the list. It's going to bring it and place it in the A site of the ribosome. It's then going to attach it to methionine. So if you look at the top of our picture, you see the yellow circle for transfer RNA and the ALA, which is alanine, on the top. If you look to the middle, you'll see them bonded together. When the two are bonded together, the ribosome is going to scoot over one codon. That's going to move alanine from the A site to the P site and push methionine out. This cycle is going to continue until the entire strand has been translated. This is another example of the same process. You see UAC, that's the P site, and CAC coming in to fill the A site. That's the second picture from the left. UAC leaves, everything scoots down, so CAC is in the P site and UUA is an empty site. The A site is empty. Its anticodon AAU comes in and fills that site, and the process is repeated. There are three codons that code for stop: UAA, UGA, and UAG. Elongation continues until the ribosome encounters this stop codon. When it does, termination begins. At termination, the completed protein, the last transfer RNA, and the ribosomal subunits will be released. The protein has been completed, and the process can begin at another point. This is the genetic code cheat sheet. There are 64 codons that make 61 amino acids and 3 codons that say stop. You use messenger RNA with this cheat sheet. If you begin with the first base as U, the second base as U and the third base as U, you create the amino acid phenylalanine. If you take a look at serine, there are 4 different codons that code for the same amino acid, and we'll talk about why that is in class. This is another visual on the genetic code. DNA is at the top, messenger RNA in the middle. It's divided up into messenger RNA codons and the amino acids that it creates at the bottom. Use this and this slide to get used to reading the code. You'll need to be able to do that for both my test and the exam. Even though the processes of transcription and translation are done very carefully, mistakes can happen. And just as in DNA replication, those mistakes are called mutations. The difference is that a mutation in DNA replication can lead to evolution and natural selection. Mutations in protein synthesis just make bad amino acids. That amino acid then can make a bad or a different protein. If the protein is very incorrect, it can lead to extensive problems within the organism. There are 2 types of mutation, a point mutation and a frame shift mutation. In a point mutation, you have a change in a single base. So if you look at the picture, the T, thymine, was changed to cytosine. Well, you know that cytosine cannot bond to adenine, so adenine must be changed as well. That basically means that you have a mistake being corrected with another mistake. When you do this, you have a base pair substitution. This base change can cause a change in an amino acid, but not necessarily. If you look at serine, there are 4 different base combinations that all lead to the same amino acid. If you look at tyrosine, you only have 2 possibilities. A change in that third base changes tyrosine to the stop codon, which can be very significant to the production of a protein. In a frame shift mutation, a single base is added or deleted. This causes each frame, which is 3 nitrogen bases, to be read differently. So each codon then is read differently, creating a different amino acid. If you look at the top, we have the correct messenger RNA and the correct series of amino acids. In the middle, we have that base pair substitution. Thymine has been substituted with adenine. That changes glutamate to valine. But as we talked before, that only involves that one amino acid. The second glutamate and the lysine remain the same. The frame shift mutation at the bottom involves the complete deletion of thymine. When that happens, all the nitrogen bases shift down. When they shift down, the codons change. And when the codons change, the amino acid changes. So glutamate becomes glycine, the second glutamate becomes arginine, and the final lysine can't even be made because we're short a base. This is why frame shift mutations are much more harmful. More than one codon is affected by that single mistake. The last section in this chapter are on chromosome mutations, and these occur during DNA replication. So technically, we should have discussed them in Chapter 14. The reason they're in Chapter 15 is because now that you know that DNA makes RNA, which makes proteins, you can see how a change in DNA then makes a change in messenger RNA, which makes a change in the protein that is made. Because they are on DNA, these mutations will involve several genes at a time. That means they involve several proteins at a time. Keep in mind that these mutations, as they are just neutral. When they become a trait in an organism, that's when they become positive or negative. There are 4 types of chromosome mutation. The The first is deletion. This occurs when part of a chromosome is left out. This happens when the chromosomes cross over unequally during genetic recombination. We will talk about this extensively in meiosis, but what happens is mom's chromosome, say number 1, matches to dad's chromosome number 1, and genetic information is exchanged. If that information is exchanged unequally, as in the picture you see genes B, C, and D are missing, that new cell is going to be missing the information for the messenger RNA and for the proteins that it makes. In duplication, part of a chromatid breaks off and attaches to its sister chromatid. This happens during replication, and this will cause a gene or a series of genes to be repeated. This can be negative because you end up with too much information and the cell can't understand what it's supposed to do, or it can be positive because you end up with extra proteins made and that double set of protein is actually beneficial. Inversion mutations are the result of two mistakes. First, part of a chromosome breaks off, and then the cell tries to fix this problem by reattaching the chromosome, but when it does, it reattaches it backward. So if you look at our picture, genes B, C, D go in as D, C, B. Sometimes this doesn't cause any change at all. If, for example, B, C, D all code for the same amino acid serine, it doesn't matter what order they go in. If, however, they're coding for different amino acids and they go in backward, we're going to break up an important gene arrangement which is going to make the protein incorrectly or not make it at all. Our last one is called translocation, and this is the worst of the mutations because it involves two different chromosomes. In this case, part of the chromosome breaks off and it's reattached to a different chromosome in a different pair. So for example, in our picture, ABC is chromosome 1, KLM is chromosome 12. When they break apart and they're reattached, we're combining information from two chromosomes onto one. Again, important gene arrangements are going to be broken up, so proteins will be made incorrectly. If those genes are two different sizes, you may end up with too much information in one cell and not enough information in another, which will cause problems in two cells or two organisms rather than one. The cell needs to regulate everything that happens, and sometimes the proteins that are coded for need to be regulated as well. This is done through DNA methylation or histone modification, and we'll be looking at these processes again later in the year. In DNA methylation, we're going to make the change directly to the DNA. Chemical tags called methyl groups will attach to the backbone of the DNA. These methyl groups, which are C and H molecules, are going to turn the genes on and off by altering the interaction between the DNA and the messenger RNA. The second type of regulation is histone modification. This is an indirect effect on the DNA. Remember, we talked about how DNA wraps around 8 histone proteins to make a nucleosome. In histone modification, how tightly or loosely that wrapping is going to be changed. If the wrapping is made to be tight, the genes within the DNA may be hidden so that messenger RNA can't see them. If messenger RNA can't see them, proteins aren't made and that gene is essentially switched off, so you can't see that gene in the organism. If the wrapping is loosened, that gene can be seen by messenger RNA, the protein can be made, and that gene will be expressed. We'll be able to see that trait in the organism. The last type of regulation is newly discovered. It was actually found in 1998, and so they're still working on this. This is called RNAi, or RNA interference. In this case, we're going to regulate gene activity in the cell, and we're going to determine whether that particular gene, the proteins that it makes, are expressed or not. In this case, the small interfering RNA molecules will bond to specific sites on the messenger RNA, and that will cause it to either make more or less of a particular protein. In our picture, the small interfering RNA bonded to the part of the messenger RNA that makes the proteins for color. And so we ended up with an increase in the production of color proteins, and so our purple proteins are enhanced, and so we end up with something that's almost black. They think that these RNAi molecules will actually be able to prevent diseases such as HIV and hepatitis. They're working on those now in the lab. And this is the end of Chapter 15, Protein Synthesis.